Welcome to the Deep Dive. Today we're taking a really granular look. We're getting into the methodological bedrock of modern cancer cell biology.
Right, the stuff that makes the science actually work.
Exactly. The protocols, the techniques, the absolute non-negotiables that underpin all robust cancer research.
And this isn't just theory. This is the operational toolkit you need if you want your data to stand up to any kind of scrutiny.
That's a great way to put it, a toolkit.
For anyone entering this field, this is essential. Whether you're a medical student running your first experiment, a professional needing a refresher, or even a self-taught learner, our mission here is to distill these core procedures so you understand not just what to do, but why all this rigor is so necessary.
And we're going from basic safety all the way to advanced proteomics and genomics. So let's start right there with rigor, foundational rigor. The sources jump right into cell line health and integrity.
And the first non-negotiable is biosafety.
Okay.
All handling of cancer cell lines, all of it, has to be done in a level IAA or higher biosafety cabinet, a BSC.
And that's for two reasons, right? Protecting yourself and protecting the cells.
Exactly. It protects you from potential biohazards, and it protects your cultures from, well, everything else in the air.
And what if you're working with a more dangerous line, say a level 2?
Then the rules get even stricter. All your contaminated media, the plasticware, anything that touched those cells has to be decontaminated, usually autoclaved before it can be thrown out.
So you have to check the biohazard level. No guessing.
Never guess. You consult the repository like ATCC. It's not optional. It's the standard of care.
Okay, so we're safe and we're sterile. But let's get to what I found to be the most shocking part of this foundation. The reproducibility crisis. The source material documents that up to 30% of cell lines used in research are either contaminated, often with the HeLa line,
which is incredibly aggressive and just takes over everything.
or they're just completely misidentified. That means almost 1 in 3 studies could be built on the wrong cells.
It's a staggering number, and it fundamentally shifted how research is viewed. You have to have proof now.
So what's the mandate?
Formal cell line authentication. That means genetic analysis, checking the cell shape, the morphology, and tracking growth curves. And you have to do this at least every 5 years.
So beyond those formal checks, what about day-to-day work in the hood? How do you stop that cross-contamination?
It's all about being meticulous. You handle only one cell line in the BSC at a time, period.
Okay.
Then, after you're done, you let the cabinet run empty for 5 minutes.
To clear the air?
To clear out any lingering aerosols that could float into your next culture. And you dedicate media aliquots. Never use one big bottle of media from multiple cell lines.
And then there are the invisible threats.
Yeah.
Genetic drift and mycoplasma. Let's start with genetic drift. What is that?
It's the slow accumulation of mutations that just happens. The longer you culture a cell line, the more it divides, and the more it changes from the original stock.
So a cell line at passage 100 is basically a different cell from one at passage 10.
Functionally, yes, it could invalidate all your data. The solution is simple, but people often ignore it. Always use the lowest possible passage number you can get.
And now for mycoplasma. The sources describe these as, well, insidious.
They're the absolute bane of cell culture labs. They're these slow-growing bacteria that, crucially, don't have a cell wall.
Which means standard antibiotics don't work.
Exactly. Penicillin and streptomycin are useless. Mycoplasma won't kill your cells outright, but they will completely alter their metabolism and gene expression.
So all your molecular data is corrupted.
Completely corrupted.
And if they're invisible, how on earth do you find them?
You have to screen with specific molecular assays. This is mandatory. We recommend testing about a week after you start a new culture, and most importantly, right before you freeze any cells down for your biobank.
Because if you bank a contaminated vial...
you're just preserving the problem for years to come.
Okay, so now that our cells are clean and authenticated, we can start building biological models. Let's talk culture environments.
Right. So we basically categorize them by how they grow. You've got your suspension cell lines.
Like blood cells.
Blood-derived cells, yeah. They grow free-floating in the media.
Yeah.
Then you have adherent cell lines, which stick to the plastic.
And those cancers, right.
Most epithelial cancers, yes. And for those, you need an enzyme, usually trypsin, to get them to detach when you need to subculture them.
And for both types, the atmosphere in the incubator is critical. Specifically, that 5% text CO2 level. Why is that number so important?
It's all about chemistry. The text CO2 creates a bicarbonate buffer system in the media. That system is what holds the pH stable.
And if the text CO2 is off...
the pH crashes, the media becomes acidic, and your cells get stressed. all your functional data becomes instantly worthless.
Speaking of function, that brings us to cryopreservation and biobanking.
Hmm.
Standard FFPE tissue is great for looking at structure, but it's dead tissue.
It is. So for functional studies, like making a patient-derived xenograft, you need to bank viable tissue.
You're capturing a functional snapshot.
Exactly. For something like PBMCs, peripheral blood cells, you start with density centrifugation on a medium like fecal pack to isolate them. Then you freeze them in a special medium with DMSO.
DMSO is the cryoprotectant, preventing ice damage. But the sources say the freezing rate is the most important step. Why?
It all comes down to physics. You can't just throw the vial in a super cold freezer.
Right.
That causes big, sharp ice crystals to form inside the cells, and they just shred everything. The gold standard is a controlled rate freezer, which cools things down at about -1°C per minute.
So it's a slow, gentle process.
A very controlled process. It encourages small ice crystals to form outside the cells, which pulls water out and minimizes internal damage. Then you transfer them quickly to liquid nitrogen.
And they can't warm up during that transfer.
Never let them warm above -50°C, or you lose viability.
So now we have viable tissue. Let's take it in vivo. Xenografts. This is where you put human cancer cells into mice.
Into immunocompromised mice, like NODSCID mice. Their immune systems are too weak to reject the human tissue, so the tumor can grow.
And if you're making a PDX, a patient-derived xenograft, straight from a patient's tumor.
It's a delicate process. You harvest the tumor, dice it into tiny 1 mm fragments, and then use an enzyme like collagenase to digest the tissue and free the cells.
And then you implant those cells with a substrate to help them grow.
Yes, you suspend them in Matrigel. It's a basement membrane matrix that gives them the structure and signals they need to establish a tumor. You keep it cold and typically inject it under the fourth nipple in the inguinal region.
That brings us to section 3, modeling the tumor microenvironment. Let's talk about co-culture using transwells.
Transwells are a clever way to study how cells talk to each other without touching. You have a permeable membrane insert that acts as a barrier.
So you can have one cell type on top and another on the bottom.
Right. They're physically separate, but signaling molecules, cytokines, growth factors, can pass right through the membrane.
And the example in the sources is perfect, studying how fat cells, adipocytes, talk to breast cancer cells.
It's a classic setup. You can see exactly how factors secreted by the adipocytes affect the cancer cell's behavior. It mimics that paracrine signaling in real breast tissue.
But to get even closer to reality, we need 3D models.
We do. In a 2D flask, a cell is just. It's not in its natural state. But in 3D culture, using Matrigel as a scaffold, cells can form organized spheroids.
Structures that better replicate a real tumor.
With real nutrient gradients, real signaling. It's a much bigger model. And you can study complex functions like invasion.
How does that 3D Matrigel collagen sandwich assay work?
It's a great model for invasion. You grow your spheroids on Matrigel, then you layer collagen I on top. It creates a barrier they have to invade through.
So you can watch them move.
And you can distinguish different types of invasion. Are they moving as a collective sheet, or are single cells breaking off and moving individually? Those have very different implications for metastasis.
Okay, let's move to high-dimensional analysis. First, immunofluorescence, or IF. On archived tissues, what's the biggest technical hurdle there?
Hands down, it's tissue autofluorescence. Fixed tissues, especially tumors full of collagen, just glow on their own under the microscope.
Creating a lot of background noise.
A ton of background noise that can completely mask your real antibody signal. You have to use quenching agents like Sudan Black B to reduce it.
And validation is key.
Oh, absolutely.
Yeah.
You need positive and negative tissue controls. You need to test your antibodies at different dilutions to prove that your signal is specific and sensitive.
Now for something truly high-tech, mass cytometry, or cytoF. Traditional flow cytometry hits a wall at around 15 markers because of spectral overlap. How does cytoF get around that?
This is where it gets really revolutionary. CytoF gets rid of fluorescent tags entirely. Instead, the antibodies are tagged with unique, stable metal isotopes.
Different atomic weights on different colors.
Exactly. The instrument vaporizes each cell and then uses a mass spectrometer to measure the mass of these metal tags. Since the masses are discrete, there's virtually no overlap.
So you can measure, 50 proteins at once on a single cell?
50 proteins. It's indispensable for studying something as complex as the tumor microenvironment. Take tumor-infiltrating myeloid cells, TIMs, They're incredibly heterogeneous.
Too complex for traditional methods.
Way too complex. But with cytometry, you can define entirely new subpopulations of these cells based on those 50 markers.
And there's a clinical example, right?
A great one. cytometry was used to find a specific monocyte population in the blood, these TexCD14 plus TexAD16 TexHLA-DR, whose frequency could predict whether a patient would respond to anti-PD-1 immunotherapy.
That's a direct link from deep phenotyping to personalized medicine. Amazing.
It really is.
Now let's switch to proteomics using tandem mass tags or TMT. What problem does this solve?
TMT solves the throughput and quantification problem. Before TMT, you had to run each sample separately, making comparisons a nightmare. With TMT, you use these isobaric tags, think of them as molecular barcodes, to label peptides from different samples. You can now do up to an 18-plex.
So you combined 18 different samples into one single run.
Exactly. You combine them and run them through the mass spectrometer all at once.
But how do you know which peptide came from which sample?
That's the magic. In the second stage of mass spectrometry, the tags fragment and release unique reporter ions. The intensity of those ions tells you the relative abundance of that protein in its original sample.
So it's scalable and very precise.
Highly. But the most common failure point is sample prep.
What goes wrong?
Detergent removal.
Okay.
You need strong detergents for protein lysis, but they interfere with the mass spec. If you don't get rid of them completely, usually with SB3 beads, before you digest and label, your data is useless.
Okay, let's move to function, starting with metabolism and the Seahorse XF instrument. What's it measuring?
It's giving you a real-time, label-free look at a cell's energy use. It measures two key things. First, the oxygen consumption rate, or OCR.
That's mitochondrial respiration.
Right, the cell's main power plant. Second, it measures the extracellular acidification rate, ECAR, which is a proxy for glycolysis.
And you can add inhibitors during the run.
Exactly. You can add something like oligomycin, which shuts down the mitochondria's ATP production. Then you can see exactly how much ATP is coming from mitochondria versus how much is from glycolysis. You can map those metabolic shifts in real time.
Now, for the sharpest tool, CRISPR-Cas9. How do you design an sgRNA to specifically target a cancer point mutation?
Specificity is everything. The key is to place the target mutation as close as you can to what's called the protospacer adjacent motif, or PAM sequence. That placement maximizes the chances that Cas9 will cut the mutant allele and not the healthy wild-type one.
And there's a newer technology that's even more precise.
Yes, base editors. They are much cleaner for single-point mutations. They fuse a nicking Cas9, which only cuts one strand of the DNA to an enzyme that chemically alters a specific base.
So no double-strand break.
No double-strand break, which means a much lower risk of unwanted genetic errors.
Finally, let's look at the computational side with RNA-seq data analysis. Can you walk us through that pipeline?
Sure, it's a systematic process. Stage 1 is pre-processing. You do quality control on the raw data with something like FastQC, and you trim off the adapters.
Cleaning up the reads.
Right. Stage 2 is upstream processing. You align those clean reads to a reference genome using an aligner like STAR, and then you quantify how many reads map to each gene.
And stage 3 is the statistical part, the differential expression analysis. Why does the software use a negative binomial model?
That is a critical statistical point. RNA-seq data isn't continuous data, it's count data. And those counts aren't normally distributed. The variance is often unequal to the mean.
So a standard d-test wouldn't work.
It would be invalid. The negative binomial model is specifically designed for that kind of count data with unequal mean and variance, making the statistical test robust. And you have to normalize the data.
Why is that so important?
It corrects for biases, like differences in library size, where you just got more reads from one sample, or composition bias, where a few super highly expressed genes can skew the counts of everything else. Without normalization, the results are unreliable.
And we still need to validate these big sequencing results with the gold standard, RT-qPCR.
Always. It's still essential because it's so sensitive and specific. But again, quality control is everything. You need to check your RNA quality. The 28S band should be twice as bright as the 18S. And you must treat your samples with DNase to get rid of contaminating genomic DNA.
And the final make-or-break step for quantification is choosing the right normalizer genes.
You can't just pick a textbook housekeeping gene. You have to test a panel of them and use software like NormFinder to select the most stably expressed ones for your specific experiment. Using multiple stable normalizers is key for accuracy.
We have covered a huge amount of ground today, from the absolute sterile foundation of cell culture all the way to complex bioinformatics.
We have.
And the main insight here seems to be that high-impact cancer research really depends on the rigorous execution of all these methods.
That's a so what, isn't it?
Hmm.
Modern precision oncology is all about capturing complexity, whether it's the heterogeneity of immune cells that you can only see with Cy2F or the subtle viability you preserve with controlled rate freezing. Failure at the method stage means you fail to see the reality of the cancer.
Mastery of this toolkit is mastery of the science.
It absolutely is.
To leave you with a final challenge then, let's put on a metaphorical lab coat. Imagine you're setting up a new lab to study the metabolic interaction between breast cancer cells, let's say MCF7s and adipocytes. You want to use a 3D matrigel co-culture system and monitor metabolic shifts.
Okay.
Name 3 distinct best practices you must implement, drawing from what we've covered today. Think about it in terms of 1. Cell line maintenance, 2. The model type, and 3. The analysis technique. What are those layers of rigor you'd need?